Hello friends, welcome in this class in Insanis Pro Biology। जैसा कि आप सभी को पता है कि हम लोग ICMR 2023 की तैयारी कर रहे हैं और ICMR 2023 की तैयारी करने में अभी तक हम लोगों ने टोटल 2022 और 2021 मतलब 2022 का टोटल 150 क्वेश्चन प्लस 35 क्वेश्चन हमने आपके साथ डिस्कस किया है, मतलब approximate 180 question from 2022, and this is the another video, and in this series we will discuss a 2021 paper, and that time we will already discuss till 69 question, not 69, 70 question. Today onward we will discuss 15 question in each video and try to cover a 2021 whole paper before your exam. First question is the material used as a bioplastic. There is a term the material used as a bioplastic means the material which is used as a plastic but biological in nature, means the material which is produced by the animal, produced by the organism, and a degradable compound. The plastic term is a self-explanatory term. Plastic is the compound, is the material which is a non-degradable, means the bacteria which are not present for any plastic material. या हम कह सकते हैं कि प्लास्टिक जो है, जब भी आप पॉलीथीन या प्लास्टिक की बात करते हैं, means there are the no bacteria for which degrade this compound. But bioplastic की बात करेंगे, the compound which is used as a plastic but the origin of this compound is a organism, मतलब the means that are the biodegradable compound, and biodegradable compound which is produced by a bacteria and archaea, is polyhydroxy butyrate. Polyhydroxybutyrate is a compound which is produced by the bacteria and archaea, and they, means the bacteria and archaea, use this compound as a carbon and energy reservoir, means polyhydroxybutyrate is produced by the bacteria and archaea, and bacteria and archaea use this compound as a carbon and energy reservoir, but this compound is also used as a bioplastic because this is a biodegradable compound, biodegradable material. The correct answer for this question is polyhydroxybutyrate. The next question is a too easy question because this is asked from the NGS, next generation sequencing, and unfortunately I will discuss this question already in a Hyderabad University. Today is a more than 2 and 3 question which is repeated in the different exam, like a Fred score is this question is asked in Hyderabad University 2022-2021 exam, and we was discuss this question very deep. The Fred, PHRED, Fred score in the next generation sequencing data is quality score. Fred score is the quality score. The option, there is only one B option which says that the quality score means the Fred score represents, this score represents a read quality score in a next generation sequencing. But this question is already I was discussed in a Hyderabad University. Means the Fred score is a quality score, and this is a Q equal to minus का 10 log 10 P. The P is the probability, means this score is also the probability based score. Then we will move in the next question, and the next question says that what does SNP stand for? SNP, in the SNP, S is the single, N is the nucleotide, P is the polymorphism. And this is a very important because this is come in the light of when we know that the some disease are due to the single nucleotide polymorphism. When we done the whole genome sequencing, then we find that the some gene are linked with the single nucleotide polymorphism in the society. The one nucleotide are changed between the two individuals, and the changed nucleotide, one nucleotide polymorphism leads to the disease, and in the some cases they lead to the lethal disease. After that, this is come in the light after the whole genome sequencing. SNP stand for the single nucleotide polymorphism. The next question is also too easy. This is called the compensation. Compensation refers to the following processes in the relation to the flow cytometry. In the case of the flow cytometry, we will use the term compensation, and this term represent, this is the process of, just remember, this is the process of correcting. This is the process of correcting for fluorescence spillover. Means fluorescence spillover correction is called a compensation. Fluorescence spillover correction is called a compensation. That is, the meaning of this is removing the signal of any given fluorochromes from all detector except the one devoted to measuring the red dyes. Means in the case of the flow cytometry, there is the multiple detectors, and that multiple detectors detect the different signal which is produced by the fluorochromes. But in the reality, in the flow cytometry, there is only one detector which is devoted to measuring the fluorochrome of the dye. So the compensation is the process of removing the all signal which is detected by the detectors except the one signal that is produced by that detector which is totally devoted for the detection of that dye fluorochromes. Means what is when we read the option, that conversion of analog signal to the digital signal, that is not correct. Elimination of overlapping fluorescent signal from the different fluorochromes, that is the correct answer. The elimination of the overlapping. What is the problem when the different detector detect the same signal? Means the signals are overlap when we saw the result. So the compensation is just removing this overlap signal, and they just take the one signal which is detected by the one detector which is devoted to measuring that fluorochromes or fluorochromes that are devoted to measuring the fluorochrome for that dyes. Means the specific detectors except the signal which is produced by that detector, they remove the another signal and eliminate the another signal to reduce the overlapping fluorescence. Okay, the correct is the B is the correct option for this question. Estimation of the level of estimation is not categorize the different leukocyte population, that is also not a correct answer. The second is the correct answer related to the compensation process. What that is not which one of the following cross will produce two phenotype in the next generation? Outcross is the cross between different two unrelated strands. That is outcross, means that not produce only two phenotype. The selfing of the pure lines, selfing of a pure line only produce the one phenotype, not a two phenotype. And incomplete dominance is also produced in the one phenotype in the F1 generation. But the test cross, in the case of the test cross, we use a T and T, and the heterozygous recessive condition. We will cross the heterozygous individual with a homozygous recessive parent, and we get this produce the two gamete T or T, and this produce the two gamete small t, and we will just draw the Punnett square, and we find capital T small t small t small t, capital T small t small t small t. Capital T small t is produce the tall, because T is the dominant over the small t, and small t produce the dwarf. Yes, sort plant. Yeah, dog plant. Means they when we done the test cross, then we find out a two phenotype. One is a tall phenotype, second is a short phenotype. Means this is the correct answer for this question. The test cross produce the two phenotype in the next generation. Okay, that is also the two easy question. The type of inheritance in which the many traits of a character are spread across the gradient and the effect of each allele in is adaptive. Okay, codominance. In the case of the codominance, there is not the each allele in the adaptive. Means codominance means both allele are dominant, like ABO blood group. In the case of ABO blood group, when A is present, then the phenotype is a A blood group. If the B is present, the phenotype is the B blood group. If the cell lack the A and the B, then the phenotype is a O. But If A and B both are present on the RBC, then the phenotype is A or B. A or B. That is the codominance. Because both allele represent its dominancy. That is not like a gradient and not a additive. So means that is not a correct answer. Monohybrid outcross. That is not also the additive. Means the Mendel's law is not additive. They shows that the one gene is a dominant and another is a recessive. When the dominant is present in the phenotype is shows by that allele, not by the another allele, and the recessive allele only represent its phenotype when they present in the homozygous condition, means the small t. And dihybrid cross is also not the adaptive nature and not spread in the gradient. Well, the polygenic inheritance means the one character decoded by the multiple gene, one character decoded by the multiple gene. Yeah, one character which controlled by the many genes, then that type of the inheritance is known as a polygenic inheritance. And the one character means color is controlled by A, B, C, D, and the every like A plus B plus C plus D, then a color is black. Means every allele, every part contribute in this part. But if we say that A plus B, then less black, and A plus B plus C means near to the black. Means in the population they find the gradient. Means if this is a high, then this is the intermediate and this is the low. We find this type of the gradient, the population. That is a polygenic inheritance. Okay, this is also the both the allele expressed themselves equally in F1 generation, except in the case of the sickle cell anemia. This if the hemoglobin is normal, then disc shape RBC is there. But if the one hemoglobin is normal but another is if the homozygous is there, if the heterozygous, then this is like this. Means, but if the HBS and HBS is there, then the cells is a sickle type. Means in the heterozygous condition they just change the phenotype. Means both allele express themselves. So that is not a correct answer. ABO blood group, just we will explain that key. When the A is present, then the blood group is A. B is present, then blood group B. If the A or B not present, then blood group O. But A and B both are present in the RBC, then the blood group is AB. Means A or B both are present in the ABO blood group. And Royan cattle is also the codominance, example of codominance. Means this is also not a correct answer. Antirrhinum majus, this is belongs to the spandrillum, and this is the example of incomplete dominance. This is the example of incomplete dominance. Means this is the correct answer because they not show the both allele express equally in a F1 generation. But except this, sickle cell anemia, AB blood group, and the Royan cattle both allele express themselves equally in F1 generation. 28. That is a too easy question. When you find this type of the question, then you just break this genotype in the small parts, like capital A small a and cross with a small a. Then capital B cross with the capital B small b, and small c cross with the capital C small c. In that condition, capital A small a, capital A small a, small a small a, small a small a. In this condition, we get a progeny capital B, capital B, capital B small b, capital B, capital B, capital B small b. In this condition, capital C, small c, small c, small c, capital C, small c, and small c, small c. Then we check that what is a question. Question is that the probability of an offspring that is a small a small a. What is the probability of that small a? The small a probability is 2/4. Out of the 4, only the 2 genotype is a small a. Means 1/2 was the probability for a small a. Capital B small b or capital B small b. Capital B small b is also 2/4, means 1/2. And small c small c also 2/4, means 1/2. The 1/2 hidden is that or AB or B, AB or C, or means plus, multiply and multiply. When we multiply this, then we've got 2, 2, 4, 2, 8. I recommend you go and watch a general science video which is title under the title of the TIFR general science. There I will discuss and or multiply rule and plus rule. Go and watch. The correct answer for this question is D, 1/8. 1/8 is the correct answer for this question. मे भी आपको समझ में आ रहा होगा. अगर आपको समझ में आ रहा है तो वीडियो को लाइक करके चैनल को सब्सक्राइब कर लो, बेल आइकन को हिट कर लो और यह कमेंट कर दिया करो. ये क्वेश्चन थोड़ा लॉजिकल है. इसमें पूछा था कि जीन फॉर द स्टार्च सिंथेसिस. फर्स्ट ऑफ ऑल, व्हाट इज द स्टार्च? इन आवर सेल्फ, ग्लाइकोजन इज अ स्टोरेज. ग्लाइकोजन जो होता है आपके बॉडी में स्टोरेज करता है. लाइक द स्टार्च इज आल्सो स्टोरेज मटेरियल फॉर द प्लांट. एंड जीन फॉर द स्टोरेज, अगर स्टोरेज करना है तो स्टोरेज किस रूप में प्लांट करेगा? तो प्लांट जो है स्टोरेज जो करता है वो सीड के रूप में करता है, ना कि पॉड के रूप में करता है. आप इसे एलिमिनेशन मेथड से हटा सकते हो. क्लोरोफिल सिंथेसिस स्टोरेज नहीं. शेप ऑफ द सीड इज अ करेक्ट आंसर, बट शेप ऑफ द पॉड का शेप से नहीं है, फ्लावर कलर से नहीं. बिकॉज a starch is a storage material and the gene for the storage synthesis is directly connected with the shape of the seed. Means shape of the seed and the storage starch synthesis gene are directly connected. This is a very interesting question. 18 number question. When a diploid female plant is crossed with the tetraploidy male, the polydy of the endosperm cell is a resulting seed. First of all, we will learn how the tetraploid endosperm endosperms is formed in the normal seed. This is the normal seed, and in the case of the normal seed, this is a podal cell, and this is synergites, and in the middle the 2n from female and 1 from male, 1 from male side come, and this will form 3n, and this is called a endosperm. When we look back this, female 2n, मतलब n or n, male 1n. Then we will consider this question. Female is a diploidy, means 2n. First meiosis occurs. And gamete form small, मतलब single n, means haploidy. But male was 4n. Then meiosis occurs, and they produce 2n pollen. The pollen have a 2n, मतलब pollen are diploidy. When we consider the endosperm female or plus male, but female contribute 2n, means n plus n, but male contributes 1 pollen, means only n, means, but in this condition the pollen is 2n, means this 2n. The total is 2 plus 1 plus 1, 2 and 3 and 4, 4n, and 4n is called a tetraploidy. Tetraploidy is a condition. When we use a tetraploidy female, if we consider, not says that this question is asked. First, the answer for this question is tetraploidy, and we will use, if the female is also tetraploidy, then after the meiosis they form the 2n, means here a 2n plus 2n plus 2n, means 2 to 4 to 6n, hexaploidy. 2 to 4 to 6 to 6n, hexaploidy, if the female is also a tetraploid. Okay. First of all, we will just revise which 7 contrasting pair of a character used by the Mendel. So when we revise that, we found plant height, tall and dwarf. Second is seed shape, round and wrinkled, seed color, yellow and green, flower position, axial and terminal, flower color violet and white, pod shape inflated or constricted. Pod color green and this side are a dominant character and this side is the recessive character. The question asks कि how many have a color base contrasting trait? Color base, seed color, flower color, pod color. 1, 2 and 3. Only the 3 characters are based on the color. Seed color, flower color and a pod color. In case of the seed color, yellow are the dominant. In the flower color, violet is the dominant and the pod color, green is the dominant. But in the case of the seed color, green is the recessive and the pod, green is the dominant. So just remember these things. This is the next question and the next question is also so interesting because this is asked that which one of the following is called a cell-right effect. Cell-right is a scientist which explain the genetic drift. When you know little about the evolution, then you know that the cell-right is the scientist which explain a genetic drift. Means fluctuation of the allele frequency by chance. Yeah, by chance fluctuation of the allele frequency in the population. This is called a genetic drift. Allele frequency not follow the Hardy-Weinberg rule. And the fluctuation is by chance. This is called a genetic drift. Correct answer for this question is C. The next question is also easy. This has come from a 12th. Bone is connected to the muscle with the help of tendon. Definitely this is memory base. Tendon. Bone and muscles are connected with each other to the tendon. The next is the easy question and this is also asked in your CSIR-NET this year, June 2023. This question is asked, but in a just a simple way. This is just information for you and this is for only confusion, creating confusion by a examiner. This is not useful for you for this question. The question said that on the genetic analysis, deletion in the maternal chromosome 15 means they says that कि 15q मैटर मतलब 15q डिलीशन हुआ है, 15 पे आपका डिलीशन हुआ है. तो which of the following मतलब which of the following can present with these symptoms? First of all, this is a question come from the genetic imprinting. This is question. This question is asked by the CSIR also this time. And genetic imprinting, there is the two condition. One, deletion of maternal chromosome 15. This is the first condition. Second condition, deletion of paternal chromosome 15. If the maternal chromosome deletion is occurs, then we called Angelman syndrome. Angelman syndrome. And if the paternal chromosomes deletion of paternal chromosome is occurred, then we called a Prader-Willi syndrome. Means the syndrome is directly depend on totally depend on inheritance of the chromosome and deleted inheritance of the chromosome. If the deletion is occur in the maternal side, then the syndrome is called Angelman syndrome. If the deletion is occur in the paternal side या parental पापा से आ रहा deleted होके, तो then Prader-Willi syndrome होगा. That is occurs because genomic imprinting is means होता है rearrangement of the gene on the chromosome. Rearrangement of the gene in the chromosome. When the gamete is formed, then the gene rearrangement is occurs, and this is called as genomic imprinting. And when the chromosomes come inside your body, and if the chromosome is come inside your body and a chromosome 15 is deleted, but that 15 chromosomes belongs to your maternal, then you suffer from the Angelman syndrome. And if that chromosomes is come from your father and this is deleted, then you suffer from a Prader-Willi syndrome. In this question they ask the deletion of maternal chromosome 15 means the syndrome is a Angelman syndrome. That is a additional information. Band 11 long arm chromosome 15. Band 11 long arm chromosome 15 are deleted in both syndrome, but the syndrome is depend on कि पैटर्नल डिलीशन हुआ है या मैटरनल डिलीशन हुआ है. That is a too easy question, but that is just memory based question. If you remember, then you are correct, or not remember, then you just mess up this question. Burkitt lymphoma occurs due to the translocation of chromosome 8 to the chromosome 14. If the some part of the chromosome 8 are translocated towards chromosome 14, then the organism suffering from the Burkitt lymphoma. Because of chromosome 8, in the normal chromosome 8 have a one gene called a MYC gene, and this gene are translocated from the 8 chromosome to chromosome 14, and this event leads to the lymphoma, and this is due to the translocation of the gene MYC from the 8 to the 14, and this is the reason for the Burkitt lymphoma. But definitely this is a memory based question. If you remember, then you will do this. You will reach for the correct answer. If you don't remember, then I suggest you just leave it. This type of the question during your exam. Thank you for watching this video. Share this video with your friend, and if you face any problem to understand any question, if you face any problem, then please drop the comment in the comment box. Please support me too, because this is for me also very hard to explain in English. Try to explain in English because my English is not too good. Bye bye.